Good evening, and welcome again to Public Perspective. I'm your host, Kevin McDermott. And tonight we'll be exploring that gray area between actual science and science fiction. Sometimes it's hard to tell the difference. But tonight we're very honored to have as a special guest Dr. Monica Laronda from Lurie Children's Hospital at Northwestern University to talk about something that sure sounds like science fiction, but it's the real deal. You and the team that you're working on have created an artificial, well, you would say prosthetic ovary that actually produces real babies, right?
We did, and thank you very much for having me to discuss this amazing research. We think it's amazing too.
I agree, by the way. I think it's amazing. That's why you're here. I think it's amazing.
So we developed a bioprosthetic ovary, and what we did was we used 3D printed technologies to develop the scaffold. And we used primary cells from mouse tissue. And what we did was we made this bioprosthetic ovary and transplanted it into sterilized mice. And what they were able to do is we were able to restore hormone function from this ovary. They were able to ovulate and produce live, healthy offspring, support this offspring with another hormone responsive mechanism, which is lactation, so with milk production. And then these healthy pups were able to go on to be adults, mouse adults, and sire or have their own children, so, pups. So it was very exciting for us.
That's astonishing. I mean, and the ovary itself have been replaced by this prosthetic device, right? So there's a million questions I'm going to ask you about that, and I may understand some portion of the answers that you'll give me. But before we get started, I'm just curious about how you found yourself in this area of research.
I think it started a long time ago when I was an undergrad, and I started working at the marine biological labs in Woods Hole, Massachusetts, near where I grew up. The coordinator's assistant that I happened to be working with was for the Frontiers in Reproduction course. And so I think that really was my first big step into reproductive biology, and now I have a Ph.D. in reproductive endocrinology.
So all of a sudden, there you were with the right tools in the right place.
Right.
And you had met the leader of the team was Dr. Teresa Woodruff, right? And did you say you had met her at Woods Hole as well?
I did. I happened to, so the Frontiers in Reproduction program had 70 to 80 faculty members that went through this course to teach at least 1 or 2 lectures, and she happened to be one of the people that I met, and of course I would have no idea that I would end up working in her lab. Surprisingly enough, I even met my future Ph.D. advisor, Larry Jameson, at Woods Hole at the same time. So I think it speaks to some of those historic labs, and just the great, amazing people that you can end up rubbing elbows with, and maybe my future destiny was that.
Well, this, in business, of course, we talk all the time about networking, and here's it is, right?
Yeah.
In practice, right? You met the right people at the right time.
Right.
So you started this research in Dr. Woodruff's lab, though, correct? Even though now you have your own lab, right?
I do.
Laronda lab.
Yes.
And I just have to say, once again, I am so impressed to be sitting with someone who has a lab with your own name on it. That's so cool.
Thank you. That was always a goal of mine before I think I realized what it took to get to that point. But now, here a few decades later, I just started my own lab in August 2016, this past year, at the Stanley Manny Children's Research Institute at Lurie Children. So I'm very excited.
And so you'll continue to work, though, with Dr. Woodruff on this, or will you, on this, whatever the next steps are in the research on the prosthetic ovary, correct?
Absolutely. So my lab is supporting this research, but I will continue to work with Teresa. She's a great mentor of mine, and her insight and expertise is invaluable. So.
Well, let's see if we can actually, there's a video that Northwestern University has put together for this that explains what actually happened. So let's see if we can get that queued up, and we'll take a look at the video, and then we'll continue with some questions afterwards. So let's see if we can get that video up and running, and then we'll talk about what we've seen.
real breakthrough here is that we're building a true ovarian bioprosthesis, and the goal of this project is to be able to restore fertility and endocrine health to young cancer patients who have been sterilized by their cancer treatment. Right now we're able to do that with young mice, and the goal ultimately is to provide this back to patients.
Teresa and I have both wanted to create an ovary bioprosthesis or some sort of implant. So we said we need something that we can mold and build for exactly this purpose. And Ramille Shah's lab was right next door.
You know, from the very beginning, with any collaboration, it's always great if it's built on mutual respect for each other's work.
Scaffolds, if you think about a scaffold for a building, they're setting that up and they're building something that will eventually support itself.
We have constructed these scaffolds out of gelatin, and gelatin is just broken down collagen. And collagen is the most abundant extracellular matrix in our body. It's what gives us structure. It's in our bones, in our skin, etc. And the great advantage of doing gelatin over collagen is that it's much easier to process, but it still has the same, what we call bioactivity as collagen.
So these illustrate the different pore structures or architectures we can achieve with our different ink platforms, as well as the 3D printer. And these pores will house the different cell types, and we're really trying to investigate how the structure can influence cell behavior and viability. And it also can expand towards other soft organ and tissue engineering applications, because we have the ability to 3D print basically any architecture that we think will be optimal for different cell types.
This is an example of one of the scaffolds that Alex has 3D printed, made out of gelatin. Our skilled technician removed the ovary of the mouse, replaced it with our scaffolds, sutured those back up, and we mated some of those animals, and we were able to get live birth out of these scaffolds.
Our hope is that one day this ovarian bioprosthesis is really the ovary of the future.
You know, there's just so much in there that is so fascinating. The idea of using, first of all, 3D printers to create organic material. And I know Dr. Shah talked about it as ink, but it's not actually ink as we'd understand it. It's it's organic material that's being laid down in this scaffolding, right?
Right. So the particular ink that we used is gelatin, and like Alex Rutz, the other first author on the paper, mentioned in the video, it is broken down collagen. So it is a bioactive material that has...
What does bioactive mean?
Yeah, it means that the cells can interact with it. So the cells perceive the receptor sites that are along the extracellular matrix proteins, and that allows them to interact with it. It allows them to break it down if necessary, just like any, what we would consider a biological material.
So tell me why this is, I mean, I can imagine, but tell me why this is so important to have this bioactive material to be able to use as the ovary. What would be wrong with, say, plastic?
Right. So especially, I think, in trying to rebuild an ovary, these points are particularly important. So the initial primordial follicles, and if I step back and just explain what a follicle is,
Sure.
it has a centralized, what we call an oocyte, or the potential egg cell. And it's surrounded by very supportive cells that are absolutely necessary for it to survive and mature and differentiate into something that would be ovulated and what we would consider a fertilizable egg. And so the structure overall is spherical. This primordial follicle will be about 30 microns, and
That's
I think
in a mouse, it's 30 microns. Right?
Well, even in a human, it's about 30 microns. So in a mouse, it will grow, expand multiple times in size. in humans, it can expand as a follicle up to 600 times that original size. So just to the size of a diameter of a penny before it's ovulated. So it just gets much larger. So plastic in this case would not work at all.
It won't expand enough. Right?
It won't expand enough. It won't be dynamic to kind of withstand that type of differentiation that needs to occur. The other thing is the cells can't interact with plastic, and they need something to be aware of their space in their environment, and they use receptors on their cell surface in order to do that. So in addition to breaking it down, they need to be able to kind of grow on it, adhere to it, things like that. So very special cells grow on plastic, and follicles and the cells of the ovary are not.
They're not
those.
Yeah. Got it. So finding the right gelatin mix must have been very difficult. I mean, given the properties that you're describing, finding the right ink, it's weird to use that word in this context, but finding that right ink, how long did it take to do that?
So Alex Rutz and Ramille Shah developed this type of ink and printing it, being able to print it in this way over a few years, in conjunction with the beginning of this project.
So they knew, when they went into it, they knew that they were trying to get this result when they started experimenting with this type of printing.
And
right?
I'm not sure that they even realized what it would be used for. And so we were right next door, luckily, and had a problem where I think their solution started to fit and take hold. So where we developed the bioplotted scaffold from that point was engineering the architecture and the design of it to be able to hold the mouse follicles.
And was that primarily a biological exercise or a structural engineering exercise to try to figure out that scaffold design?
I think it was both. So we, in the Woodruff lab, we looked at the extracellular matrix, the natural extracellular matrix components of the ovary, and we are very, we have cultured ovarian follicles from mice and from other organisms, even in humans, in different biomaterials. So we know what they like and how to kind of maintain that shape. But the previous methods that we were using were encapsulation, and so at the very end where ovulation needs to occur, they were kind of trapped inside. And so the biomaterial needed to be removed. So many different collaborators have gone down the road of trying to look at encapsulation and modify those type of biomaterials to be able to be digested and things like that by the ovary. But what we wanted was something that was more translational and something that could be transplanted in a way like the bioprosthetic ovary that we ended up producing.
So did the one you produced, that did it follow a different path from the encapsulation path that the other researchers had been looking at?
The other researchers were able to obtain live birth from encapsulation methods of ovarian tissue. We're looking more at some ways to isolate follicles and to individually seed them, and then put into something that's scalable for human use. And I think getting back to your question, I think it was both a biological and an architectural or engineering feat. It was definitely a research technician, Callie Whelan and I's trial and error from a different lot of, for a lot of different architectural designs that Alex would print. And she would vary strut, which is what we call the strand, coming out of the printer. She would vary the thicknesses of that, the distances, the angles.
How many layers?
How many layers? We ended up with a solid bottom, which was actually key to making. I don't know. There was just some way that made that work as far as seeding the follicles on more than an empty bottom.
And I love that, by the way, that that you don't necessarily know why that works, but you discovered that it does.
Right.
Right, and that seems to be so much a part of the scientific method that you don't always know why, but
Right.
here, this one worked and the other one didn't, so let's go with this, right?
Exactly, and there was a lot of trial and error in the lab, and I think the good thing with a 3D printer is that you can kind of go back to the drawing board fairly easily, change the program slightly, and try 3 new iterations of a scaffold for Kelly and I, to try again.
So how many, was there hundreds of iterations, thousands, tens of thousands? How many iterations did it take to get one that
I would
worked?
say dozens. It was yeah, it definitely wasn't hundreds. I mean, like I said, we started at least with an idea of what we needed.
From studying the actual
From
ovaries.
studying the actual ovaries,
Okay.
right.
Yeah. So the design process was working from a real model that everyone knew worked. Can we get something close to that? Now I would imagine though that it's, you can't actually, maybe you can, but it doesn't seem likely that you could actually replicate the actual design of the real ovary. You probably come up with something that is a little different but that suffices. Is that correct?
That's true, yes. So when we look at the extracellular matrix or the organ skeleton, how we look at that is we removed the cells in a process called decellularization. So you remove all of the cells, and it's kind of a popular method for regenerative tissue engineering and things like that. And so a paper prior to this that Teresa and I published looked at the organ skeleton and the extracellular matrix and how that's formed and what it's made of and what the pore design looks like around the follicles and things like that. What we found was that the follicles were already kind of there and built that extracellular matrix, of course, around themselves with the help, of course, of the stroma and the other cells of the ovary. So then our goal became to plan something that could be digested and flexible and dynamic like gelatin and respond to the cells that we've put in there and kind of allow them to restructure it. Give them enough support that they'll survive and grow and be happy and then, but not so much that they're restricted. And so that's another way that I think we described in the video, using like a scaffold, like a building, and allowing the cells to kind of do their thing.
Which seems only reasonable. I mean, the cells have been doing this for millions of years.
Right.
And they kind of know what they need to do.
Right.
So you're just providing the environment in which they could do it.
Right.
Now, one of the things that in reading about this, it was mentioned was that there's no rejection of this ovary, even though it's a foreign body that's being put in. And we think about organ transplants, and rejection is one of the biggest problems that's always faced. So how is that overcome by the fact that or how did you overcome that?
Right. In this case, we used a mouse with a special genetic mutation that has a suppressed immune system. So it's the equivalent of being on immunosuppressive therapy. So it's very similar to what people have in transplants. However, I see this being slightly different in practice once we hope to translate this into the clinic. And that's because we will be testing the gelatin scaffold or whatever type of scaffold ends up working for human follicles and make sure that itself doesn't have some sort of immune trigger an immune response.
Right.
But we hope to be using the patient's own follicles or the own potential egg cells and their own steroid-producing cells, hormone-producing cells. So we're really hoping that type of transplant they wouldn't reject their own cells.
Right, and the scaffolding itself, the gelatin is something that's essentially non-interactive. I'm not sure what the right word would be, but it wouldn't trigger an immune response, the gelatin itself. The scaffolding?
That definitely has to be
tested,
yeah.
Yeah. So there's still a lot of work that yet to be done, right? Yeah. So we talked a little bit before the show about any possible ethical concerns, and there was an article, as we discussed in The Economist recently, about an artificial womb
Right.
where sheep were being, they could spend the rest, for premature babies essentially, who could spend the rest of their time developing in this artificial womb and then be born in a normal schedule and be healthy and whole, essentially.
Right.
So as we think about these things, think, well, now we have an artificial ovary and an artificial womb, and at some point, are we going to need humans to grow humans? Is that something that you guys think about? Do you, in your team, or is there an ethical team that you have to confer with?
Right. So Teresa established the Oncofertility Consortium, and with part of this in mind. So what she is an advocate for patients that might lose their fertility and hormone function because of their oncology diagnosis,
Right,
for example.
Right. Like radiation would be something that would kill ovaries, right? Yeah.
Right. And many chemotherapies. Yeah.
Yeah. Okay.
So as part of that team, she has tasked ethicists and other types of researchers and clinicians and patient advocates to think about this. And we think that it's an important ethical question to think about in the future. I think where we are and what we are hoping to achieve, at least in our first iterations of a
Right.
bioprosthetic ovary,
Right.
they would be using their own genetic makeup and their own potential egg cells. So this would be something where they would be passing on their own genetic traits in this case, and are still definitely needed to participate, required to participate.
So the Brave New World scenario is, maybe
far.
we can see it from here, but it's still
Right.
a long ways away, right?
But you're right. There are ethical questions to consider. I mean, we're talking about something that could produce life, but the human cells are still definitely involved.
Yes. Yeah. Good to know. Yes.
Yeah.
So it's one of the things that comes up a lot about science and scientific research, and in another field that I'm familiar with, artificial intelligence. There's this question about where does the artificial intelligence lead us, and would AI ever be a threat to humans? And so it's very easy to go down the path and think this is really wonderful, because in the case of people who through cancer treatment have lost the ability to have children, now you restore that ability. That's a wonderful thing, and no one would argue. But it opens the door to other things, right? So is this something that's a normal part of the process, like in your own lab, where these kinds of ethical things are kept as not necessarily in the forefront, but at least within eyesight?
Absolutely. At least in the labs that I've been involved in, that has definitely been the case. And there are, we work directly with the hospital. I'm hired by Larry Children's Hospital. And we work at, there's Northwestern Memorial Hospital as well. So we're constantly within the guidelines of things that represent not just humane treatment for our animals, but, but considering ethical treatment of human patients.
Yes, as a human patient, I'm reassured by that.
Yes.
It's nice to know. One of the other things I noticed about this team is that it's all women, which I think is also really cool. We're so used to thinking of scientists as, TV and movies all have scientists as, guys with gray hair like me and, white lab coats. And in your case, it's all women in this particular team. That's fascinating. Is that by design, by accident?
Definitely by accident. I mean, there's two middle authors on our paper who are males, so we're not opposed to it. But you bring up a great point. It just was very coincidental that I think all of us who are experts or who are working to solve this problem happen to be female, and it made it made it fun and interesting, and I hope inspiring to other girls who see the type of work that we can come up with, that they're not all men. Yeah.
Well, and I would think that because it's reproductive health, men don't think about that nearly as much. So it doesn't surprise me, actually, that you would be more focused on it. But I also want to say, yes, I like to think of your team as being role models. And it's interesting to hear you think that way as well, that you can see your team as providing that kind of role. Do you do any kind of outreach to students to think about this kind of thing?
We definitely do. Teresa is a great advocate for this, and so as part of the Oncofertility Consortium, she has other aspects of outreach for young girls. They're high school girls, they come in, and so being in her lab and being a part of that, you know, teaching them and showing them what bench research looks like, and even in some cases what mice anatomy looks like.
Sure. They may never have
really
seen a mouse. It was not the inside of a mouse.
Right.
So,
yeah, on the street doesn't count.
Yeah,
It's
okay,
a little
gotcha.
different. Yeah, so that has been really rewarding and definitely a great thing to have as part of the lab culture.
Mm-hmm.
To have both boys and girl high school students, but in particular, she does outreach, particularly for young girl high school students. So that's been really exciting because I think sometimes you do need a role model. It's easier to see
Yeah.
yourself in that position
If you're seeing someone else who looks like you.
Mhm,
doing it. Yeah.
exactly.
I've spent time and interviewed a number of people in education where that comes up a lot, where people have to see someone who looks like them in order to really get the realization that, oh, I could do that too, right? And I think that's important.
Yeah.
So let me segue quickly from that though. You talk about scaling, and I think of scaling in a different sense. Like if we're looking at this kind of process to create artificial ovaries, how far are we from being able to 3D print an artificial lung or an artificial heart?
Right. Well, I can't put a timestamp on that, of course. But I think what we have shown, which is the first restoration of a soft tissue organ in an animal that I know of. I think the way that it's designed, the way that we've thought about this process, using cells and kind of giving them the environment to do their thing, could definitely be translated to other organs. I mean, in fact, you know, the beta cells in islets in the pancreas, for example, that produce insulin, are spherical, so that even that shape itself would translate pretty well. So I think this is just the beginning, and I think that we could scale it to different organs, definitely. Yeah.
If not your lab, then others.
Right.
Right.
Right. I'm focusing on reproductive endocrinology and trying to restore fertility for our patients.
So what is next for you? Now that you've had this major breakthrough, what's the next step?
So we hope to continue with this work, in particular, in larger animal models. We're hoping that the success in mice will translate into larger animal models and translate for human follicles. So we do have some research tissue where we could use human follicles to test those initial experiments and that design of the 3D printer. But using larger animal models is definitely something that's more representative of human physiology and anatomy. Would be, you know, even a greater breakthrough in my mind if we could get that to happen.
And then, of course, eventually there's humans as well, right? Well, on that note, believe it or not, we're out of time already.
Oh, wow.
So when you make that next breakthrough,
Yeah.
we'll have to get you back and we'll talk about where you are in the course of your progress. So thank you so much for being a guest here. It's been wonderful talking to you, and I've learned so much.
Thank you. I've really enjoyed it.
So thank you. We've been visiting with Dr. Monica Laronda, and I'm Kevin McDermott, your host on Public Perspective. You can see us every Saturday night at 8 on Comcast Channel 19, and you can find us on the web at publicperspective.tv. So till next time, thank you and good night.